Assalamu alaikum, dear students. I am Ftir from MS Chemistry first semester. The presentation topic is Fourier transform infrared spectroscopy. First of all, we will discuss about the introduction. Fourier transform infrared spectroscopy is a powerful analytical technique used to identify functional groups in molecules based on their absorption of infrared radiation. It works by measuring the absorption of infrared high light by a sample and processing a spectrum that provides information about the chemical bond present. Fourier transform infrared is widely used in chemistry, biology, material science, and other fields for qualitative and quantitative analysis of organic and inorganic compounds. Now we discuss the principle of Fourier transform infrared spectroscopy. Fourier transform infrared spectroscopy is a technique used to analyze the infrared absorption or emission spectra of a sample. It relies on the principle that a sample absorbs infrared radiation at specific wavelength characteristics of its molecular structure. The Fourier transform aspect refers to the mathematical process used to convert the raw data obtained from the instrument into a spectrum that represents the sample's absorption characteristics across a range of infrared wavelengths. This transformation allows for high resolution spectra to be obtained efficiently, providing detailed information and about the chemical composition and structure of the sample. Now we will discuss about the instrumentation. Fourier transform infrared spectroscopy is a powerful analytical technique which is used to identify and characterize chemical compounds based on their absorption of infrared radiation. Here is a breakdown of key components and operations of an Fourier transform infrared spectroscopy. First of all, there is a source. Ftir spectrometer typically use a broad spectrum infrared source such as globular silicon carbide rod or heated filament to emit infrared radiation across a range of wavelengths. Interferometer. The heart of Ftir spectrometer is an interferometer such as a Michelson interferometer which split and recombines the infrared beam. This allows for the measurement of the sample absorption of infrared light across a range of wavelengths spontaneously. Third instrument is a sample component. A sample component holds the sample to be analyzed. Sample can be in various forms. It may be solid, may be liquid or gas. The component is designed to minimize their interference for its external source and to maintain a controlled environment. Now, it is a detector. Modern Ftir instruments use sensitive detectors such as liquid nitrogen, cold mercury, sodium tolerate detectors for polyelectric detectors. To convert the infrared signals into the electrical signals, this signal is processed and used to generate the spectrum data system. The interferogram generated by the Interferometer is converted into a spectrum using Fourier transform. This digital signal processing technique allows for rapid and accurate measurement of the sample's absorption spectrum across a wide spectral range. Software. Dedicated software controls the instrument data and processes the resulting spectra. It also allows for spectral analysis, including peak identification and qualification of functional group or compound present in the sample. Accessories. Various accessories can enhance the capability of an FTIR spectrometer, such as attended total reflectance cells for solid samples, for liquid cells, for solid samples, and gas cell for gas samples. These accessories enable analysis of sample in different physical states. FTIR spectroscopy is widely used in fields such as chemistry, pharmaceuticals, material science, and environmental sciences due to its versatility and ability to provide detailed chemical information about samples. There is an overview of the principle. You can observe it. There is a mirror, broadband source, beam splitter, delay line, mirror, sample detector, interferogram, FTT, and spectrum. How? There is a graphical interpretation of FTIR. Infrared spectroscopy is a technique which is used to study the vibrational mode of molecule. The graphical interpretation of the Fourier transform infrared spectroscopy typically use a plot of absorbance or transmittance, wave number or wavelength. Here how it generally looks. Absorbance for wavelength. Observance usually plotted on the x-axis. It is represented how much of the incident infrared radiation is absorbed by the sample. It represents how much of the incident infrared radiation is absorbed by the sample at each specific wavelength. Wave number plotted on the x-axis. It is measured in reciprocal centimeters and corresponds to the frequency of the infrared light absorbed by the molecules. There is some key features. Absorption band. There is a pair of or peak or trough on the spectrum. Each band corresponds to a specific vibrational mode of the molecule. Baseline. The baseline represents the background absorbance or transmittance level of the solvent or sample holder without the sample. Position of peak. The position along the x-axis indicates the frequency of vibration associated with different chemical bonds within the molecule. Intensity of peak. The higher intensity of the peak correlates with the amount of absorbance at that particular wave number, which in turn relates to the concentration of the absorbing species. The types of vibrational mode may be stretching, asymmetric, and symmetric stretching. Vibration appear at high wave number around 280 to 4000 centimeters. Bending vibrations appear at the lower wavelength about 500 to 5 centimeters. Now, there is some advantages of FTIR. Improved sensitivity. FTIR provides higher sensitivity due to the ability to collect and process a broader range of frequencies, simultaneously requesting in enhanced signal to noise ratios. Speed. FTIR can acquire a full spectrum rapidly because it measures all frequencies simultaneously, whereas dispersed method scan sequentially. Multiple advantages. FTIR allow for multiplying where multiple frequencies can be measured spontaneously. This enhances data speed and accuracy resolution. FTIR often achieves higher spectral resolution because it utilizes longer path length and higher precision optics than dispersive techniques. Accuracy and precision. The digital nature of FTIR data and processing reduce errors and allow for more precise measurement and analysis. Versatility. FTIR can be used for a wide range of sample types for gaseous to solid and can handle samples in various physical states like liquid, solid and gases. Quantitative analysis. FTIR facilitate quantitative analysis through techniques such as peak height or area measurement, which more straightforward to the digital nature of the data. Overall, Fourier transform infrared spectroscopy have revolutionized infrared spectroscopy by offering faster, more sensitive and more accurate measurements across a broad range of applications in chemistry, in biology, material science and more. There is some limitations. Complex interpretation. It requires expertise to interpret spectra correctly. Sensitivity to water. Water can interfere with the analysis due to the strong absorption in the infrared region. Dear students, thank you so much and Allah Hafiz.